Okay, so we finished talking about mitosis on the last lecture. So make sure that you guys kind of review that a little bit. As a quickie review, I'm going to ask you what of these things fall under mitosis. Does anaphase fall into mitosis? Is prophase a part of mitosis? Telophase? Metaphase? Interphase? No, that's right. It is not part of mitosis. What's happening in interphase? Growth is in G1 and G2. S in the middle there is when we do what? DNA replication, right? And then what about cytokinesis? Where does that fit in? We kind of tack it on to the end of mitosis because that's where we end up separating the actual cytoplasm for our two cells. So it starts in telophase. And what would happen if only one of the daughter cells received all of the mitochondria, for example? The other one would die because it has nothing to make ATP, so it can't do any of its processes. And Jeanette, you had asked a couple lectures back about exocytosis, endocytosis being an active process. Yes, it's an active process. It needs ATP to be able to do it, even though we weren't showing you that in there. So let's move forward on to protein synthesis. So we've briefly mentioned this story before. You'll hear me repeat it multiple times. And if you think of it more of a story, it's easier to try and remember what's going on. So if we look at the nucleus of our cell, and where is DNA housed? Just in the nucleus. And when I am copying my DNA, but making it into its photocopy of RNA, we call that transcription. So if you think of like when somebody's like a court stenographer and they're typing what everybody says, they're transcribing, they're copying down everything that you say, right? That's why this is called transcription. We're going from DNA to RNA. They are both a nucleotide language, if you will, right? Because it's telling us what nucleotides belong in what order. And then from there, depending on the order that it's in, it will correlate to certain genes. And so when we do DNA to RNA, notice it says here pre-mRNA, right? Pre-mRNA is what's made first. And then we need to modify it a little bit, change it up a little so that it can now be able to be protected when it goes out of the nucleus to the cytoplasm. Because what's out in the cytoplasm? Nucleases, which can break down our RNA. And so what we'll do to the pre-mRNA is it actually has a lot of A's attached to it, so we put fake ends on either side of it so that when the nucleases, if they start getting on and attaching and eating things off of there, taking apart our nucleotides, it hasn't gotten to the actual message or recipe that we want. Okay, so we'll put the fake ends on it, and a lot of times we'll put tons of A's or adenines at an end. And we will also have what's called a start and stop codon on that messenger RNA. Okay, and it does just what it sounds like. It's going to tell us in a minute where the ribosomes need to start reading the message and when they need to stop reading the message. And that's what will end up saying this is the beginning of the protein, this is the end of the protein. Here you go. So when we go from, after we've made our pre-mRNA, then we modify it and we get our mature or messenger RNA ready. That then leaves the nucleus through what? Through the nuclear pores, and it goes out into the cytoplasm. Okay, so in the cytoplasm, it's going to meet up with ribosomes. There's going to be a large ribosomal subunit and a small ribosomal subunit. They kind of stick together. Large on top of small, and then that's what's actually going to read your messenger RNA. If it's too loud, you guys can close that door if it's too noisy out there. When we go from RNA to a protein, we call that translation. So you can think of translation as if RNA is a nucleotide language, and then we're now changing it into an amino acid language, which is what the protein building block is, right? You can think of it as somebody translating English to Spanish. So that's why it's called translation. We're going from a nucleotide language to an amino acid language. So we have transcription occurs in the nucleus, and translation occurs in the cytoplasm, right? All right, a couple other things that you guys know from this picture is if it's a double-stranded helix, it's a DNA molecule. If it's single-stranded, it's RNA. If you see T's, you know you're talking about DNA. If you see U's, you know you're talking about RNA. Awesome. So what else do we need to tell you about this? Also, DNA is never by itself. That's why it's always double-stranded. What shape or look does the DNA have in order for us to do translation or transcription and then translation? No? Sorry? Stretched out and in the chromatin form, right? Chromatin versus chromosome shape. Okay, both are DNA. It's just a matter of how tightly coiled it is or relaxed. And when it's relaxed is when we can read it and do things with it. It means we're actively using the DNA. Let's see. All right, so terminology. And don't hesitate. Go back and forth on this picture and try to talk about that story without, having a whole bunch of notes on there, just so that you could look at that picture and understand what's happening, right? You know what the story is. So gene is a segment of DNA coding for one polypeptide chain. So for example, if we have our DNA strand here, it could have multiple segments on it, which it does, and one segment here would be gene number 1. This would be gene number 2, gene number 3, gene number 4, gene number 5, so on and so forth, however long that strand of DNA ends up being. So if we take that segment, we're going to look at different parts of what we would do. That gene would read off for one messenger RNA, for example. That would be one recipe. That would be the code for maybe the protein for hair color, or the code for the protein for eye color, or skin tone, or your height, or different things like that, okay? So DNA is read in groups of 3s. We call it a triplet on the DNA. But when we transcribe it to RNA, that triplet code is something that we call a codon. So codon is associated with RNA, okay? So codon specifies the placement of a single amino acid. So if we had, for example, A, C, U, G, A, U, G, C, C, A, A. And we have our covalent bonds in between, right? And because we see U's, what molecule is this? RNA. It's RNA. And if we were to break it up into triplets, we would see ACU would be gene number 1. GAU would be gene number 2. GCC would be gene number 3. AAA ends up being gene number 4, for example, right? So you could figure out whether what the DNA molecule was from this, right? Because it's a complementary strand, you can always figure out, if you're given RNA, you could figure out what the DNA strand originally said. If you were given DNA, you could tell what the RNA strand is going to say, all right? And you should be able to flip back and forth and know which kind of molecule you're looking at and how to determine the other one. So a typical protein will have about 100 to 1,000 amino acids. Okay, so if one amino acid is coded for by the 3 letters that give you the codon. So one codon gives you, which is a pair of 3s, gives you 1 amino acid. Okay, so 3 times 1,000, right? 3,000 RNA nucleotides together is what had to give you that. But I didn't tell you, we also chop it up and move things around so it folds right and things like that. So it's actually going to be longer than it actually needs to be. We modify it some more, and then you get your real, true protein that gets used and utilized. So there are actually 3 types of RNA. we have ribosomal RNA or rRNA, mRNA, which is messenger RNA, and we have tRNA, which is transfer RNA. And so we need all three of these types to make our protein, okay? The rRNA or ribosomal RNA is the workbench for protein synthesis, and RNA is made in the, it's in the nucleus, but even more specifically, within the nucleus is the nucleolus. And we briefly mentioned it when we were first looking at the cell, right? And let's see. And if we draw the RNA, we had our long or large segment. We had a small segment. It goes out into the cytoplasm where it's going to bundle itself up, and that helps protect it from nucleases. And then you have your small unit right there, and that is your rRNA molecule. Messenger RNA is the copied code for the protein. So copied code for protein, okay, which originated from DNA. That's how it got transcribed, right? And it's going to go from the nucleus to the cytoplasm. And mRNA is where we're going to find the codon. tRNA, or transfer RNA, does what it sounds like, and we'll see it in detail in a moment, is going to add the building blocks which are what? The amino acids to the growing protein chain as it's being assembled. And it's on the tRNA that we will have something called the anticodon or anticodon. Anticodon. So mRNA has the codon, think of it as the actual code, and tRNA will have the anticodon on it. Okay. So there are two main steps in protein synthesis. We have transcription and translation. So with transcription, transcription is, again, what process? Copying DNA to RNA. It occurs in the nucleus. Okay. So this here is what we would call like a transcription bubble. Okay. And in copying down, you can see the double helix has gotten pulled apart. It still is there, right? We're not getting rid of the DNA because we don't want to damage our original, our template strands. So we read off this piece of RNA. So this is our mRNA that's being made. That was our DNA up there, okay? And that's an enzyme that helped unzip it, helicase, and then it allows you to keep the hydrogen bonds from forming again, so you keep the strand apart while you make your RNA. When we no longer need this gene being read, the gene on the DNA, then what happens? The DNA closes back up. It'll zip itself back up. It'll reform its hydrogen bonds. Those are easily made, easily broken, right? And so we'll get the signal like with insulin. When we have enough insulin, there's no more glucose in the bloodstream. We no longer need more insulin. Then we will say, hey, we don't need any more of this. Shut down, stop making your messenger RNA. So we stop making the protein for insulin, and there you go, right? And then who goes and stops the RNA from being read over and over and over again? Nucleases will eventually chew up the RNA. Right. So, okay, so going back to transcription. So transcription is making mRNA. Okay. And translation. So they're kind of showing you on this picture here. You have this DNA molecule, and they're saying this little segment here is gene 1, this segment here is gene 2, this is gene 4 over there. So this top strand right here is what type of molecule? DNA, because it has what? It has thymines in it, right? So remember, triplets are on the DNA strand. Then we have our codons. Codon tells you it's a what strand? Messenger RNA. You have your codons there. And so those are your sets of 3 that we're reading. We also know it's RNA because there are uracils on there, right? We see U's. And then notice that so we already knew that RNA is a complementary strand of DNA, right? If you look, the tRNA, which is the bottom molecule here, tRNA, notice that it's a complementary strand to the mRNA. So where you had an A, you make a U. Where you had a U, you have a A, and where you have a G, you have a C. Okay? And one thing that I want you guys to notice is that this is the one codon you have to memorize is AUG. AUG is the start codon. You don't have to memorize all the other codons that tell you things like which amino acid it codes for. But the codon AUG, and we know codon means messenger RNA, codes for the amino acid methionine, or MET. Okay? All proteins start with this. This is what says, put a methionine on, this is the start codon, now you know when to start reading this molecule off and being able to, this is where the protein starts, okay? So, AUG means what? It's a codon and it codes for, it's a start codon and it codes for the amino acid methionine. Okay, the only other codon that you have to memorize are 2 of the stop codons. There are a few more, but UGA or UGA is a stop codon. And the other one is, I'll find it, we'll come to it. All right, well, UGA is one of them, okay? And notice, is there an amino acid associated with UGA? Nope. There's no amino acid, but it tells you stop, that's the end of the protein, stop sticking amino acids on, okay? So, another way you can remember AUG as the start codon, AUG is like August, school usually starts in August, so that'll help you remember that, right? Ooga is a weird word, you can remember that, right? Ooga, ooga, stop, something like that. Whatever it takes to remember it, right? Okay, so the only ones that you have to memorize is AUG is the start codon that's found on messenger RNA. It codes for what amino acid? Methionine. And what's one of our stop? MET is the abbreviation for it, but methionine is METHIONE, methionine. Oh, NINE. Yeah, methionine. Okay, and so let's go to our diagram. This is the same story that you guys saw before. So diagram of translation. So transcription happened in the nucleus. We moved on to translation. So you have the start codon is always AUG in every protein produced. And the stop codons that you have to memorize was UGA, which was UGA, and here's the other one, UAA. Those are the 2 stop codons you need to memorize. There are others, but these are the only 2 I want you to memorize. Okay. And this is what tells you to stop putting it on. And if you kind of read through the story here, they're showing you a little bit of transcription inside the nucleus. When it comes out to the cytoplasm, so here's your messenger RNA leaving the nucleus into the cytoplasm. It's going to meet up with the large and small ribosomal subunits. So that's this big pink thing here, large and small, and this was your mRNA. Notice on your mRNA, that's where you have your codons, your start codon AUG, right? So it meets up with a transfer RNA. Now within the large and small ribosomal subunit, there are 3 sites, and you can think of it as EPA, like Environmental Protection Agency, or if you want to read it the other way, APE, whatever works for you. But E means exit site, P is the actual placement where we're building and making the actual protein. A is kind of like the holding pattern, okay? So the ribosome is coming along reading this, and what it does is it says, hey, I need the tRNA that is my complementary code to my codon. So what was the tRNA called that was the complementary code to a codon? Anticodon. So this is the anticodon for AUG. It was UAC. It's just the flip, right? You guys can do that. And the tRNA is kind of like this weird clothes hanger shape here, okay? And on the end, it's got methionine attached to it. So that little circle there, or that bead, is our amino acid. So this goes into a little more detail than you guys need to worry about, but initiation just means this is where it's going to start doing things. Elongation, just like what it sounds, is you add things on, you're elongating the chain, you're making it longer. And you can see in this lower picture here, they're showing you that the next amino acid here is coming to the A site getting ready, because it knows what's going to be next, right? So notice on the P site where you have your anticodon and the tRNA, and see how our chain is getting longer and longer, and there's nothing in the E site. So the empty tRNA ends up moving to the E site, and then it'll get ejected, and it will go find another amino acid. Does it find the same amino acid or a different amino acid? It will be the same because it's always whatever it's coded for. It will always be coded for methionine on that particular tRNA. The other tRNA that has like leucine or proline or one of the other amino acids on it will always code for that one because that's what its codon is for. So when it gets released and it now no longer has an amino acid attached to it, it will float around, go out into the cytoplasm and find another amino acid that it will pick up, the one that it is in particularly coded for, and then it will come back and deposit the next amino acid. So it keeps dropping off amino acids, getting empty, looking for another amino acid, coming back, dropping it off when it's time in that chain, right? Yes?
So is it kind of like a lock on and a key? It'll only accept?
Kind of, yes, because it will not code, nothing else can attach there except for the amino acid its anticodon is coded for. Okay. And so that's why it can't attach anything else to it. And if you didn't have any of those amino acids, then you're not going to have anything to put on there, right? You're not going to be able to finish your protein. It's waiting, going, hey, where's the next one? How come it's not here? So it's partly why it's essential. We have certain amino acids that our body cannot make on its own. We have to get in our diet, basically. And if we didn't have them, then you start seeing deficiencies in people, right? Okay. So, and it will keep reading. It'll know when to start because it'll see what on the codon? The AUG start codon, which puts on which amino acid? Methionine. So it'll keep going until it hits the what? The stop codon, either UGA or UAA, right? Those were the two I want you guys to know. And when those come into contact, so here's your UGA, right? And notice there is no amino acid attached to it, right? It has what's called a release factor, and it says, okay, we're done making our protein. You can now let go. This protein gets released, and now it's ready to go to either parts inside the rough endoplasmic reticulum, or it might go into the Golgi at that point where it will then get folded up, and we might clip off some ends and change things a little bit, package it to get it ready to go outside of the cell. Maybe it's a protein we need within the cell. So it just depends what it's needed for, who is this protein, and where is it going to go next. So that's what we call termination, is when everything is done, we got our stop codon, we use a release factor, and ta-da, you are good to go. All right. And the main thing is to know that the P site is where you are actually doing elongation. And each time, if we kind of come back up to this picture right here, so what happens is when you get an amino acid attached, okay, you have to break the bond here between the tRNA to the amino acid, and you've got to create a new polypeptide bond or a new peptide bond, or that's the more specific name for a protein's covalent bond, right? You have to create a new bond there, but if we don't let it go, then it's stuck on there, right? So you're breaking a bond from the tRNA to the amino acid while you're attaching that amino acid and making a new covalent or peptide bond so it attaches to the chain, okay? And if we look at the term polyribosome, let's see. On polyribosome, so as long as AUG exists, you will have another ribosome attach and make another protein. So here we have where it says it's the start of mRNA. So that's where our AUG codon is on the messenger RNA. And a ribosomal large and small subunit will come together and start reading and making its protein. And then when it gets to the stop codon, it lets go, and the large and small ribosomal subunit will let go, and then they will find another messenger RNA to attach to and start reading. And while that's on there, nuclease can't attack the mRNA, correct? Because it's protected when the ribosomal subunits are on there. And so it will continue to do this over and over and over again until you finally get the signal, your homeostatic balance, right? You'll get that signal that says, hey, I don't need any more protein right now, we're done. So then eventually what will happen is when you're no longer making more mRNAs, there is less of it out into the cytoplasm, and eventually the nucleases will start to attach on there and be able to start chewing off the ends. Once it chews off like the A of the AUG, what happens? You won't know where to start making a protein, so you'll stop making it, and then ribosomal subunits don't know where to attach anymore, and that's where nuclease will start chewing it up. And it doesn't matter when we get rid of the messenger RNA, because if we still needed it, we'd be making more in the nucleus, right? Making more DNA to messenger RNA and keep making that until we no longer need it. And it doesn't matter when mRNA gets chewed up, because we either continue to make more of it until we no longer need it, or we can now break it down because we have met our needs and we no longer need to make that protein anymore for that time being. And then it gets chewed up and we can then recycle the nucleotides again, right? So that's that in a nutshell. Let's see. So translation occurs where? I tricked you. Translation occurs in the cytoplasm. Transcription occurs in the nucleus. Good, good, good. All right. So let's take a look at extracellular materials, if I can. Oh, we're at the bottom of the notes. That's why it won't cycle. Okay, extracellular materials. These are substances found outside of the cell. So this is everything but the cell, right? If it's everything outside the cell, everything but the cell. So with body fluids, we have extracellular fluid, which is all fluid outside of the cell. Another example would be like blood or cerebral spinal fluid. If we look at interstitial fluid, this is specifically between body cells. So this is between cells. And plasma, for example, is the fluid part of blood, and CSF is cerebral spinal fluid, where we find in the dorsal body cavity, right? Is interstitial fluid always extracellular fluid? Yes. Yes. Is extracellular fluid always interstitial fluid?
No.
No. Why?
Because it could be around the cells, not necessarily between them.
Exactly. It could be around the cells and not necessarily between them. So for example, plasma or cerebral spinal fluid. Okay. If we look at extracellular matrix, that is protein or polysaccharide substances that help hold the body together. So you could think of this as material. It's kind of like a jello type of material or fibers. Let's see if I can fit this on here. Jello type of materials or fibers. Okay. So an example would be collagen fibers, for example. All right, let's bring up chapter 4. So chapter 4, tissues, the living fabric. So hopefully some of this will be a little bit of a review. You guys also now have your lab manuals, finally came in yesterday. So make sure you guys get over there and pick them up if you didn't already. And this will also make more sense in your lab manual, having this lecture going along with it. So tissues are groups of cells similar in structure that perform a common function. So you have been looking at the 4 main types of tissue. What are they? Epithelium, muscular tissue, nervous tissue, and connective tissue. Right, awesome. Okay, so epithelial tissue. There are 2 types, coverings and linings, and glandular. So coverings and linings are on free surfaces of the body. That means there is always air next to one of the sides of the epithelium. And we use the term the apical surface oftentimes when we're talking in lab. So an example would be skin surfaces, or also lining internal cavities. I'm sorry? Lining of the lungs? Yeah. So lining any cavity, and even the tubes into the lungs, the bronchioles, that type of thing. So, or lining internal cavities. And then the other kind, glandular, forms glands of the body. And if you guys noticed on looking at some of the glands already in histology, they were very cellular. They had lots of cells bunched up around them, right? So special characteristics of epithelial tissue, the cellularity. You didn't know that was a word, did you? Cellularity consists of closely packed cells, which you guys have seen. They have specialized contacts, which we've already talked about. So cells fit together to form sheets. What did we call these? We call them cell junctions, desmosomes, and tight junctions, right? Okay. And then with polarity, they actually have a polarity. They possess 2 different surfaces. So we have the apical surface and the basal surface. Apical surface is the upper or free exposed surface. So sometimes when you guys were looking under the microscope, you were in the middle of the tissue and you couldn't find that apical surface. And you were kind of going, what am I looking at here? And I said, well, find the apical surface. You know, go back out to lower magnification, find some outside edge, then look there and work your way in from there. And if you're not on the right edge, go to another edge and come in. Yes?
With the open side, so, okay, so if you're in a tube, which is the apical, the inside of the tube?
Everything facing into the tube would be the apical surface of it. And yes, and I'll show you a picture. So the more medial or inner edge away from that apical surface would be the basal surface, or you might find the basement membrane there. So yes. So basal surface is the lower attached surface. And so they're just showing you a picture here, apical surface versus the basal. And apical, just remember, is always air on this side, always. And again, that air could be in a lumen. We're using air as a generic kind of term, right? So blood vessels, the lumen side of it would be the apical surface of it. The other side of it would not, okay? Supported by connective tissue. That makes sense, right? After you had the epithelial, you then had something underneath it usually, which was CT. Different kind of tissue type, right? And so we'll talk about that in the middle, in a minute. Avascular but innervated. What does that mean?
No blood but nerves.
Exactly, no blood but nerves. So avascular means no blood. A something means no something. So avascular, no blood within, but it does have nervous tissue. So I just put a positive sign, positive for nervous tissue. Okay. And also regeneration, a high rate of cell division. What cell division? Mitosis, right. So mitosis, and it means it's actively occurring. And it makes sense that some cells will need to regenerate faster than others, especially cells that are more on a surface versus maybe cells that are don't have a large function to it. But the cells, for example, like within our mouth, might get abraded more often than some of the cells on the outside of our skin, right? So depending, even within epithelial tissue, there might be different rates of what's going on. So let's look at classification of epithelia. This should be a good review for you guys. The classification is by shape of the cell and the number of cell layers. So squamous means, in Greek, it meant Latin for scale, so scale-like. Scales on a fish, thin, that type of thing. So there are the three basic forms: squamous, cuboidal, or columnar. Squamous is thin and flat, and if we draw like a side view of it, Sherry likes this one. It kind of looks like a fried egg, where the nucleus would be like the yolk of it, if you will. And so this area right here would be the basal surface of it. This area up here would be the apical side. Okay. And notice that it's flat and thinnish. If you look at cuboidal, it means it's never taller than they are wide. Which makes sense. If you have a perfect square, it's equal, right? And to me, they look like poofy square balloons, basically. Okay. Columnar makes sense. They're going to be taller than they are wide. And if you look at them, they're column shaped. They're kind of a long rectangle, right? So if we classify them by numbers of layers, if we use the term simple, simple means single layer. And what it means with single layer, they're usually going to be for filtration and absorption areas. So filtration or diffusion of substances. So you're usually going to find these within capillary walls, walls of air sacs, and we give it a special name when it lines, when it's the lining of a blood vessel, and that's called endothelium. Endo, kind of like within, right? Thelium. Okay. And they also line the cavities around a glomerulus. So this picture over here is what you guys have in the lab manual. And it was easy to identify a simple squamous cell there because it's really thin. And so, for example, here's one squamous cell, simple because it was a single layer. Here's another one right here. Here's another one. Notice how squished and thin it is. My circle is big, but the cell itself, I'm just circling where the nucleus is. So then we asked you to look at the lining of the cavity around a glomerulus in the kidney. And when you guys looked at those cells under the microscope, what did you see? Lots of cells. You didn't see this big open space here, right? Because that was for the lung. That's why we wanted you to see the kidney cell, because it was a little harder to identify the simple squamous cell there when it had a bunch of other cuboidal cells and other things around it, right? So also lines cavity around a glomerulus, which is in the kidney. So for example, lung tissue will do filtration and diffusion of gases. Let's see, one of the things that Sherry was showing you guys too is if you drew the glomerulus in this space here, then you had something within that was lined by simple squamous cells. So that was the glomerulus here. I'll put G for glomerulus. And then you had simple squamous lining that area. And then you had a bunch of cuboidal cells around that. And that's where you guys sometimes got lost when you didn't know which type of cell you were looking for, right? So if we look at simple cuboidal, single layer of our squishy cells, right, our big round cubey balloony cells, you can see here, for example, here is one cuboidal cell, right? In the center of this area here is thyroid hormone, for example, if we're talking about the thyroid, which I think was what you guys were looking at in lab possibly. And then usually simple squamous, you can't see the cytoplasm. So when we go back to that previous picture, can you see a bunch of cytoplasm in there? It's so thin and flat, you didn't really see it, but you could see cytoplasm in these cells because you could see the nucleus, you could see the cell membrane, right? And you saw a bunch of stuff around it. That was the cytoplasm. So if this is your cell, here's your cytoplasm, there was your nucleus, right? Also on here, this area right here would be the basement membrane for these cuboidal cells. So here's one cell here, here's another cell here, here's another cell here, another one there. But that line there is our basement membrane, or the basal surface. Whereas into the center here, with this glandular tissue, for example, and we're producing something that gets secreted into that luminar duct area, right? Why do we have some cells there have a nucleus in it and some did not?
Because just the way that it was sliced.
It was the way it was sliced, yeah.
Part of the nucleus on some, but others.
Exactly. Some of the nucleus on some of those cells either got chopped off in the way that it was sliced, so you don't see a nucleus. It had one at one point. There's only one cell that doesn't have it, right? Which is erythrocytes or red blood cells. So that's why some of these you saw nuclei, some of them you did not. And if you were to, say we're doing the histology eval, and you had to pick a cell to put your pointer on for simple cuboidal, stick it on one that's really obvious, has a nucleus in it, the most beautiful one that you can see there within your field of view, right? Because it's an eval, it's a test. Pick the best one, right? So don't pick one without the nucleus. Let's see. If we look at simple columnar, okay, with simple columnar, notice how they're column-shaped or elongated, right? So, let me go back up to cuboidal. Sorry for a second. So simple cuboidal a lot of times is for secretion. So this would be glands and the wall of ducts. And the other function they do is absorption. An example of that would be kidney tubules. Okay. If we look at simple columnar, that would be for absorption. That's a P. And secretion, especially in the digestive tract. Okay. So in this example, can you guys see where the basement membrane would be and where the apical surface is? So this would be the apical surface, and this line right here would be your basal surface or basement membrane. Okay. And sometimes it's not always easy to see because the cells underneath it are a similar color from the staining, but look at the shape of the cells. If the cells look a lot different and go from the apical surface down, that will help you identify it easier. And then pick where you think it looks like, oh, this is a change in how the cells look. This is the end of that area right here. Here's my basement membrane for those particular cells. So in the stomach, for example, we'll secrete hydrochloric acid. In the small intestine, we're oftentimes absorbing nutrients. So that's why you're seeing digestive tract, you're seeing absorption, you're seeing secretion, that type of thing, okay? Characteristics unique to columnar epithelium. So some of you guys have had to find, and hopefully you all tried to look for, goblet cells. And once you started to find them, they were a lot easier to see, right? And notice that when you looked at different slides or different areas, some of them were more roundy, some of them were a little more elongated. But where did you see them near? Near the apical surface. With columnar, simple columnar epithelium, right? So goblet cells are for mucus for protection and lubrication. So they secrete mucus for protection and lubrication. So easy to find in the trachea and digestive tract, right? What was the job of microvilli before? Increased surface area for increased absorption. So increased surface area for absorption. Oftentimes found in the small intestine, which makes sense, right? We're doing a lot of absorption of nutrients there. And with cilia are formed by microtubules, remember? So microtubules form cilia.
So
Yes.
you can't really see the microvilli on the simple columnar, can you?
You may be able to, it just depends. It may just look a little fuzzy to you. It may be a little more obvious. It might depend on the staining technique, or if you go to 100 X and use oil immersion, you might be more likely to see it than just under 400 total magnification.
How do you distinguish from the cilia?
It'll look different. Cilia is more hair-like, and the microtubules are going to be the invaginations of the cell membrane. So they are different looking, yes. And if you kind of look at this picture down here, you can see cilia on there. It's very hair-like looking on those cells. Yeah, try 100x on one of the cells that may have microvilli, and so look in the small intestine and then see if you can see it that way. And might have to be even something where they see it more on electron micrograph, where that's even more powerful magnification. They're bombarding it with electrons to be able to see it. And it looks like a grainy picture when you see those in the books, or if you Google it, you'll find some pictures, and then you can come back and see if you can see it this week on the microscope possibly. So the cilia, microtubules form the cilia, and these are found in the oviduct, trachea, and these move things along the surface of the cell. Okay. All right, pseudostratified columnar epithelium is always ciliated. And an example would be in the trachea. And we can see some goblet cells on here too. Do you guys see the goblet cells? There's one. There's another one. You guys are used to seeing them look more whitish a lot of times. But just keep looking at different slides. You'll see different shapes. You'll see different staining. And pseudo, of course, means it's a false stratified look to it. So if we drew like a picture of columnar cells, you might have where they kind of look like they're in different shapes. But it's really a single layer, one basement membrane, the apical surface. Let's see. So in the trachea, a lot of times you'll cough things up like mucus, and cilia have a one direction on the way that they push things. So they'll relax, and then they propel things across the surface of the cell. And then it'll relax, so it keeps pushing it in one way. It wouldn't make sense if it went both ways. For example, trachea, you get a water droplet or mucus there. You don't want that getting into the lungs. You got to move it out. So it's going to be a one direction push of things across the surface of those cells. Let's see. And the other thing too is all of the pseudostratified epithelium are touching the basement membrane. So no matter what weird shape it had, they all were touching the basement membrane, each cell was. And stratified epithelia, so that means 2 or more layers of cells named for the shape of the outer row, where it's most durable. So if you kind of look at both the cartoony picture and the actual cell picture here, notice that near the basement membrane, the cells look like a different shape than when they get towards the top or the apical surface of the membrane. The newer cells are going to be down at the basement membrane, and as they age and get older and new ones get formed underneath it, it pushes them further and further towards the surface, okay? So the stratified squamous is for protective areas. So surface squamous, deeper, they're going to, on the more basilar surface, they're going to look a little more cuboidal or columnar, but they are named for what the apical surface looks like. So that's why it's stratified squamous. So let's say at the surface, they look squamous, so that's why they're going to be called stratified squamous. But deeper on the basal surface looked cuboidal or columnar. So they are usually 20 to 30 rows thick. And most widespread of stratified epithelium would be the skin and into openings from the skin. So into the mouth, lining the anus, lining the vaginal area, the urethra, things like that, okay? So skin and openings from skin. Okay? So outermost layers are often dead. Why? Yep, pretty much. Nutrient diffusion. So they have less access to the nutrients, which is more prevalent down at the basilar surface where they're younger and healthier. And then eventually there's still a protection, though, even if they aren't as healthy and young and poofy, right? And eventually they slough off. We shed skin, just not quite like a snake or a lizard. So example areas would be tongue, mouth, pharynx in the throat, urogenital areas, and these are all high abrasion areas, and that's why it's okay to lose the scales. But if we damage, like on the basilar surface, it takes a lot longer to heal because now you're having to grow all those layers of the cells back, right? So, and if you think about it in your mouth, when you eat food, you're scraping things on the inside of the mouth. Your teeth, the food, you eat like a corn chip and it like scratched you, things like that. It's high abrasion areas, right? Keratinized means that they contain keratin, which is waterproofing protein, but we like to think of it more as water resistant. So what it really does is it's protecting us from losing our water, okay? So it keeps the fluids in us. Do we need keratinized cells in our mouth? No, it's pretty wet in there, right? We have saliva, we've got all kinds of wet stuff in there that we're secreting all the time. So we don't have it on like the inside of the mouth and probably not so much linings of the body cavities, right? But where would we need it? On the epithelium that's on the surface of our skin. Right. So better protection and stuff. So this is going to be skin only for keratinized. Let's see. Stratified cuboidal and stratified columnar are both rare. So we don't usually see multi layers, but you'll see when you do see it, it'll just be 2 layers deep, a few at the most. And we don't even have pictures of it because it's so rare, so don't worry about that. But examples of stratified cuboidal would be ducts of larger glands, ducts of larger glands. So for example, sweat, mammary, saliva, salivary glands. Okay, those would all be stratified cuboidal. Stratified columnar would typically be found at transition zones. Bless you. So this is where we have a changing of the epithelia. So for example, in the male urethra, at the beginning it's more of a stratified squamous. Then as we get into the male urethra a little bit further along, it becomes stratified columnar. And then if we get further along, it then becomes transitional epithelium. So it's a change in the shape of the epithelia as it's going along, depending on where it's at or what's going on. So male urethra is a good example. Male urethra. Okay. When we look at transitional epithelia, so that's this picture down here, this forms the lining of the urinary tract. So lining of urinary tract, where there's going to be a lot of stretching, right? So it's a surface layer where it looks more domed shape. Can you guys see how more fluffy bulbous looking it is at the apical surface there? So dome shaped at the surface when it's not stretched. And what do you think it looks like when you have a bunch of fluid in there putting pressure against it? Kind of flattens it back out, squishes it down. So this would be a relaxed, not full bladder, for example, right? So always associate transitional epithelium with bladder. Okay. And the outer cells may be binucleate, which means two nuclei. So, for example, here's one of those binucleate cells at the top there. Okay. So, basically, with if it needs additional help, more of a control center, extra help telling it to do things, that's where you'll have additional nuclei within the cell. Skeletal muscle will have more than one nuclei. Most of our cells are just uninucleated or one nuclei per cell. So it goes to more of the needs of the cell and what its functions are, and that it might need more oversight and control of two things, kind of two heads are better than one sometimes. I don't know. So what epithelium is best for wear and tear that needs waterproofing, keratinized, and stratified squamous? Stratified because it'll have multiple layers. It doesn't matter if it gets kind of rubbed off a little bit or abraded, right? Let's look at glandular epithelium. So this is single or multi-cell structures that secrete a product or secretion. So two cell types, either cuboidal or columnar, all right? So cuboidal or columnar. So we have two main glands we're going to talk about, endocrine glands and exocrine glands. Endocrine glands secrete directly into the bloodstream. They are ductless and they produce hormones. And you guys have seen hormones before. What are the two areas that you guys have seen hormones? Lipid-based and protein-based. If it's lipid-based, it's going to be cholesterol or the sterone titles, right? And otherwise, then it's protein-based, okay? If we look at exocrine glands, they are the most common type. So these contain ducts. All other glands are these. So anything we didn't mention is now an exocrine gland. Examples would be sweat glands, oil glands or sebaceous, salivary glands, and these guys secrete, their secretions are delivered to a surface, internal or external. So secretions delivered to a surface, external or internal surface. Okay, so exocrine are the most common type, all right? We have unicellular, which means a single cell gland, okay? So if it's a single cell, there is no duct, right? It secretes mucus. A good example would be a goblet cell, right? You guys have seen them. So goblet cells. So also to the surface of respiratory tracts or GI tracts, right? That's where we're seeing our goblet cells. If it's multicellular, meaning many cells, they will possess three components. They will have a duct, a secretory unit, and they will have supportive CT or connective tissue. So these surround glands, and can divide gland internally. And we would call those lobes. And if they're small lobes, we call them lobules. L-O-B-U-L-E-S, little lobes. And the purpose of that would be to help bring in nutrients. So we will classify them by the actual duct. So we can have simple, which means it's single unbranched duct. Single unbranched duct. If it's compound, it means it's a branched duct. And we can also classify it by the secretory unit. So that would be tubular alveolar or tubulo-alveolar. Okay, so if we come back up and look at the picture here. So simple meant a single duct, right? So here's your single duct, and the pink part here is what's talking about the secretory unit in here. So if it's tube-shaped like this, then that's why it was tubular. And here, this one was branched, so it's simple because it had one duct, but it's branched tubular because it had 1, 2, 3 different branches or tubules on there, okay? If it's an alveolar shape, it's the grape-like clusters of secretory sacs, so they're more roundy looking. So it's still simple because of what? There's only one duct. And it's branched because you actually had multiple alveolar shaped sacs, the more roundy shaped secretory unit. This one's compound, so it still had, so in this case it has multiple ducts. So here, here, and here. And we had multiple alveolar secretory units. Okay. And if we look at tubulo-alveolar, both tubular and alveolar structures. So some of these are round and some of those were tubule shaped, see? So compound because we had multiple ducts going on there, and tubulo-alveolar because it's tubular and roundy. An example of that was a salivary gland. Let's see, what else? Modes of secretion in the exocrine system, right? So we're still talking about exocrine here. So we could have a merocrine. So these function through exocytosis. So the cell is not destroyed in this process. So cell not destroyed probably means we're going to learn about one that is, right? So cell not destroyed in process. It is the most common type, merocrine. And an example would be sweat gland. Okay. So the contents are released from a vesicle inside of the cell that fuses with the cell membrane and then allows us to secrete it out. Okay. As opposed to a holocrine one, this is where the cell ruptures. So that means due to a buildup of product, it's then replaced by underlying cells. So due to a buildup of product, and it's replaced by underlying cells. An example would be an oil or sebaceous gland. So basically the apical cell, the ones on the apical surface, okay, ruptures. If it ruptures, it means what? It died, right? So the product buildup in the apical cell causing the cell to rupture. The cell is then replaced by underlying cells. So you can see here's your basement membrane or basilar surface, and it grows and moves up. And then notice how these guys are bursting and releasing their contents, and then that will then come out of the pore. So this is how oil or sebum is produced. Cells are pushed to the surface. The product is released to the lumen and kind of creeps out or seeps out. So with sweat glands in the skin, like your forehead, your hands, and your feet, you guys sweat there. Oil in the hair and on the face. This would also be the process if somebody has acne and it's getting a buildup of it and then it finally ruptures. So let's see. And so holocrine kills, the cell ends up dying because it ruptures, right? All right, let's look at connective tissue real quick. So now we're in CT. We were talking about epithelial tissue. Now we're talking about CT. This is the most common of all tissues, okay? So we have different cell types. We have a macrophage here. What was another name that we called it generically? Phagocyte, right? So there's our macrophage. We have fibroblasts, so that helps build fibers. We have lymphocytes, which are for infection. We have fat cells or adipocytes, right? We have mast cells. Anybody find that in lab yet? Well, guess what? Now you know where to find them. They are only in loose connective tissue. So now you know where to look, right? So loose connective tissue, this helps, or it actually causes a histamine release. So you're going to see a lot more if somebody's having a histamine reaction. They become allergic or sensitive to something. They start releasing histamine. It's because of the mast cells. Neutrophil, this is our white blood cell cleanup. We also have what's called the extracellular matrix. We have ground substance, which is all the other stuff in the area. We have fibers, so we have collagen, elastic, and reticular fibers. Collagen is the most common fiber. Elastic fibers are made of the elastin protein. Reticular fibers are collagen fibers, but they are thin and you can't read my writing. We'll come back to that. Capillary. So variable, sometimes they are present, sometimes they are not present with the CT. So let's stop there for now, and we will pick up with common characteristics of CT when we come back. And I'll have you guys take out your